Introduction to Food Science and Technology, FST, Part 2. Define term chromatography? Chromatography is a dynamic physical process in which molecular components in a mixture are separated because of their different affinities for two substances referred to as phases, one being fixed or stationary and the other mobile phase. What are the types of chromatography? 1. Liquid chromatography. 2. Gas chromatography. Enlist modern separation techniques? 1. High-performance, precision, pressure liquid chromatography, HPLC. 2. Thin layer chromatography, TLC. 3. Gas chromatography, GC. 4. Atomic absorption spectrophotometer, AAS. 5. Flame photometry, FP. 6. Fourier transform infrared photometer, FTIR. What is HPLC? HPLC is high-performance, precision, pressure liquid chromatography. It is a separation technique used to separate non-volatile compounds and high molecular compounds, e.g., antibiotics. Define the term gas-liquid chromatography, GLC. Gas chromatography is a technique for separating volatile substances by percolating a gas stream over stationary phase as liquid, so-called gas-liquid chromatography. Briefly explain spectrophotometry. UV-vis light absorbed by the sample as a function of wavelength and radiated in the form of light spectrum which is detected by photocathode tube and the signal is recorded. What is thin layer chromatography, TLC? TLC is a separation technique in which compounds are separated between two phases, namely stationary phase and mobile phase. Define the term lipolysis? The breakdown of fatty material into free fatty acid and glycerol, with the heat of lipolytic enzymes, called lipolysis. Define the term proteolysis? Proteolysis is the term that describes the breakdown of proteinaceous material. Briefly explain the term fermentation. Fermentation is the anaerobic or partially anaerobic oxidation of carbohydrates. During this process, enzymes elaborated by microorganism break down carbohydrates or carbohydrate-like material into substances that are less subject to undesirable microbial activity than the original material. Give the names of industries which are included in fermentation. Baking, brewing, wine making, vinegar manufacture. Enlist the fermented foods: yogurt, cheese, sauerkraut, pickles. What is alcoholic fermentation? Explain with equation. In this fermentation, sugars are converted into ethyl alcohol and carbon dioxide with the help of several yeasts, most common according to this equation species of genera: Saccharomyces, C6H12O6, glucose, greater than 2C2H5OH plus 2CO2. What is acetic acid fermentation? Acetic acid bacteria, species of the genera Acetobacter and Acetomonas, are capable of oxidizing ethyl alcohol into acetic acid and water. C2H5OH, acetic acid bacteria, greater than CH3COOH plus H2O. What is lactic acid fermentation? Lactic acid bacteria utilize sugars present in the foods and convert them into lactic acid. C6H12O6, lactic acid bacteria, greater than lactic acid plus H2O. Water hardness and treatments. The hardness of water is measured in parts per million or in grains, with onegrain equivalent to 0.064 grams of calcium carbonate. Soft water contains 1 to 4 grains per gallon of some organic matter and has no mineral salts. Hard water contains 11 to 20 grains per gallon. Water activity, water activity, or AW, is a ratio of the vapor pressure of water in a solution, Ps, to the vapor pressure of pure water, Pw, A equals Ps, Pw. Water binding capacity. The ability of a protein to bind water. This ability depends on the number of charged and polar groups along the protein chi. Bound water. Water that cannot be extracted easily. It is bound to polar and ionic groups in the food. What's meant by bound water? The water which is physically or chemically bounded, which can be or cannot be removed. It is unavailable as solvent, and it is also non-freezable. Maillard reaction, Maillard-Browning reaction, non-enzymatic Browning reaction involving a reducing sugar and a free amino acid group on a protein. Caramelization is the development of brown color and caramel flavor as dry sugar is heated to a high temperature. Chemical decomposition occurs in the sugar. Solution or dispersion, two-phase system with a solid dispersed phase and a liquid continuous phase. All-purpose wheat flour, white flour, not containing the bran or germ. Combining the properties of hard and soft wheat. Bleached flour. Bleaching the pigment to a whiter color naturally by exposing pigmento air or by chemical agents. Bran. The layered outer coating of the kernel, offering protection for the seed. Germ. The embryo. The inner portion of the kernel. Endosperm. The starch-storing portion of the seed that produces white flour and gluten. Bread flour. Made from a hard wheat kernel, with a high protein-starch ratio, high gluten potential. Cereal. Any edible grain that comes from cultivated grasses. Roots, underground. Beet, carrot, jicama, parsnips, radish, rutabaga, sweet potato, turnip, yam. Sweet potatoes are a yellow to orange color flesh, either dry or moist. In the U.S., they may be known as yams, and both names are stated on a label. The non-orange, true, yam, is yellow, white, or purple pigmented flesh root vegetables. Tubers, underground. Enlarged fleshy stem. Starch storage area after leaves manufacture carbohydrates. Buds or eyes form new plants. Irish potato, Jerusalem artichoke. Bulbs. Stems with an underground bulb of food reserve. Garlic bulb, leeks, onions, shallots, spring onions, green onions or scallions do not possess aerial bulb. Stems. A plant's vascular system, nutrient pathway. A lot of cellulose. Asparagus, celery, kohlrabi, rhubarb. Leaves. The manufacturing organ for carbohydrate which is then stored elsewhere in the plant. Brussels sprouts, cabbage, lettuce, parsley, spinach. Also seaweed and greens, such as beet, collards, kale, and mustard greens. Flowers, clusters on the stem. Artichoke, broccoli, cauliflower. Fruits, the mature ovaries with seeds, generally sweet, and fleshy. Apple, banana, berry, and orange. But including vegetable fruits such as avocado, cucumber, eggplant, okra, olive, pepper, pumpkin, snap beans, squashes, and tomato that are not sweet, but contain seeds. Seeds, in fruit of a plant, may be in pods, includes legumes such as dried beans, and peas, and peanuts, and in the United States sweet corn. Although it is a grain, not vegetable, may be sprouted. Carotenoid. The group of red-orange pigmented fruits and vegetables. Some are precursors of vitamin A and also have antioxidant value. Nutraceuticals. The name given to a proposed new regulatory category of food components that may be considered a food or part of a food and may supply medical health benefits including the treatment or prevention of disease. A term not recognized by the FDA. Phytochemicals. Plant chemicals. Natural compounds other than nutrients in fresh plant material that help in disease prevention. They protect against oxidative cell damage and may facilitate carcinogen excretion from the body to reduce the risk of cancer. Hydrolysis. Breaking of one or more peptide bonds in a protein to form smaller polypeptide chains. Hydrophilic. Water-loving. Characteristic of polar and charged groups. Hydrophobic. Water-hating. Characteristic of nonpolar groups. Protein primary structure. Specific sequence of amino acids along the protein chain, joined by peptide bonds, the covalently bonded protein backbone. Protein secondary structure. Three-dimensional arrangement of sections of the protein chain. Secondary structures include the at helix, B-pleated sheet, and random coil. Protein quaternary structure. The noncovalent association of protein chains to form a discrete unit. Protein tertiary structure. Three-dimensional arrangement of the whole protein chain. The shape that a protein chain assumes in space includes fibrous and globular structures. Collagen. Connective tissue protein. The largest component that gives strength to connective tissue is solubilized to gelatin with cooking. Connective tissue. The component of animal tissue that extends beyond the muscle fibers to form tendons which attach the muscle to bones. It connects bone to bone. Endomysium, perimysium, and epimysium connective tissue surrounds muscle fibers, muscle bundles, and whole muscles, respectively. Rigor mortis. Postmortem state 6 to 24 hours after death in which muscles stiffen and become less extensible. Onset of rigor mortis correlates with depletion of ATP in the slaughtered anima. Candling. Viewing the inside and shell of an egg by holding it up to a bright light. Emulsifier. Material that allows two ordinarily immiscible substances to mix. Foam. Increased volume of beaten egg white that holds shape as protein coagulates around air cells. Gel. A two-phase system where liquid in a solid that have properties ranging from soft and weak to hard and tough. Protein digestibility corrected amino acid score, PDCAAS, a measure of protein quality that compares the amino acid balance with requirements of preschoolers and corrects for digestibility. Used by the FDA for labeling and by the WHO. Thanks for watching.